The next type of structural protein that we're going to talk about are microfilaments, and these are involved in muscle contraction, cell migration, and cellular movement. Actin is a very abundant protein in eukaryotic cells, and once it's synthesized, it folds into this globular shaped type of protein, and these molecules can bind to ATP. So you can see that here, ATP in blue, and then once it's hydrolyzed, it turns into ADP. So that's part of this process of attaching to this positive versus the negative end here. So that's defined by ATP versus ADP hydrolysis. So the structure of that globular protein changes. If it's G-actin, that means it's free actin. It's not polymerized yet. And then once it's attached and that's hydrolyzed, ADP, that structure changes slightly, and there are two different types. So this is the F filaments, which microfilaments, which is they're bound and tethered onto this chain, and then G is actually the free actin. Actins are involved in multiple different types of function within a cell. Various types of actins are found in different places. So muscle actin actually is alpha actin, and these are part of bigger, thicker filaments. And there's also beta and gamma actins, which are part of thinner filaments, and these are typically involved in different regions of the cell, non-muscle actins, for example. We'll talk about this muscle part first when we look at what's called striated muscle, or muscle with the tissue has a striation, which means that there's kind of this little groove, and it has an interesting organization of this muscle tissue that we'll look at right here. So if we're looking at a muscle cell, what's actually interesting about muscle cells is they become polynucleated. So the plasma membrane between cells as they're kind of developing goes away, and this becomes one big cell with multiple nuclei. And then you can see here mitochondria in there, but the big component of this are these myofibrils, or these muscle stacks of these actin sarcomeres. So if we look down, we kind of zoom in from this nuclear cell, we can look into this kind of stacked area. That is actually made up of different groups of what are called these sarcomeres. And we have these actin filaments on the outside, and then thick myosin filaments in the middle, which is all, again, made up of these actin molecules. And so then you can see what's happening when we have a contraction. These little kind of arms are kind of pulling on this filament, and it's causing these purple filaments to pull together. So that's the contraction that's going to happen during this process. Here, this is where myosin filaments and the alpha actin filaments work together to use this change in shape of the myosin to change the shape of this muscle fiber. That shape change that's happening when we have those little arms kind of pulling, what's happening is these are actually binding calcium. And the calcium is stored in these areas here that are the sarcoplasmic reticulum. So the sarcoplasmic reticulum is the same as the endoplasmic reticulum. It's given a different name because it's associated with these sarcomeres. So it's in the muscle, but it's holding calcium in it. And then when we need to contract our muscles, they release the calcium into that space, and then that causes the structural change and the contraction. And then that calcium's absorbed back up, or it's transported back into the sarcoplasmic reticulum to start that ability to contract those muscles again as well. So here, mitochondria, there's different sets of these myofibrils stacked together within the muscle cell. So this is, again, one cell that's made up of all of these different components as well. Here we're using dyes just to kind of see those different striations. So this is the different tracks of the cells here, and you can see the sarcomeres and the different components that we were showing you in the cartoons, how these are set up and stacked so that they can use those different kind of alternating actin and myosin filaments to kind of pull on each other and contract as well. So that's just some different ways of some different imaging techniques to use to look at these stacks. But you can see what I mean when I say striated muscle. it's got these little tracks of different kind of pigments that kind of show us where these different sarcomeres are. In this image right here, we can see the I band that contains only the alpha actin, and the A band here is going to contain the thick myosin microfilaments. So these are kind of different areas that are containing some of those different proteins that we're talking about. There are, for the other type of the non-myosin types of muscle types of actin are involved. They can be organized differentially depending on the types of proteins, the actin associated proteins that are bonded to these. So they can help organize them in either kind of stress fibers that adhere to the substratum of the cell, these bundles, and they can also help, they're also contractile actin bundles as well. So we can move the cell and change the shapes subtly throughout that cell type. We have different stress fibers, cell cortex, lamellipodia, or filopodia. So different regions, depending on where we are at the leading edge or within that growing edge, that can be branched. And these are all dictated by the types of proteins that are associated with these actin molecule. Here's a scanning electron microscopy image that kind of shows you some of the actin filaments in a filopodia are collected into this bundle, and then they're released, and it's kind of a different network that's organized a little slightly differently as in this leading filopodia. These branched edges down here help to support some of the structure of the rest of the cell. As I alluded to earlier, there are associated proteins that interact with actin, and this is an example of some of these compounds that will bind to sequester alpha actin. So we want them in the area, but we don't necessarily want them forming filaments. For example, thymosin beta 4, this can bind to the actin monomers and keep them from polymerizing. There are other types of associated proteins. Here is one that looks like kind of little funny arms. Formin, this can actually help to polymerize and activate the formation of longer polymers of these filaments. And you can see how this might use, if we have these formin dimer binds and associates with a growing filament, and then it basically grabs those subunits and kind of places them on this growing filament to help to elongate that actin. Other actin-associated proteins can be involved in, as I mentioned, the structure. And here we can have things that can branch, that allow us to branch off the polymer. So this is a nucleation site from one of the growing polymers. Looking at this ARP23, and WASP actually activates the ARP, and then it is attached to the middle of this actin microfilament, and then on that it can attach and cause the formation and the polymerization of another kind of not exactly perpendicular, but 70-ish degrees coming off of that. And you can kind of see here electron microscopy showing you some of those different branch sites off of actin. There are other types of compounds that will actually cap and stop the polymerization, stop the growth on that leading edge. For example, this capped protein will terminate that. So there are different types of proteins that are involved in changing the shape, changing the structure, the overall organization of these things, the growth, the breakdown as well. So CAPZ is one of those capping proteins, filament capping protein here that you can see would stop the polymerization of that end. There are also compounds that will cleave and cut in half the actin microfilament, and that would be gelsolin, for example, one would be this purple kind of group that would cut those growing polymers in half to kind of stop the growth or reduce the growth on the back end. What this breaking apart can do is it then can create new plus ends for different growth in different directions and different types of organization and branching. And then there's also different types of cross-linkers. So different proteins that stabilize the organization of multiple sets of polymers, either if they're going perpendicularly or parallel with each other. So filament, for example, helps with this more perpendicular organization, and then alpha actin, fibrin, those are going to make more parallel. So it depends on if we're kind of in the leading edge of the phylopodia, for example, some of those longer projections, or we're kind of in the basal area of the cell within the cell. Microvilli are really important structures within our intestinal cells, and they are actually going to be really important for increasing the surface area of the cell. So you can see this big long projection. They're really made up of actin protein bundles. So we have the negative side, positive side, the leading edge of growth here. And you can see those fibrin, those bundles, those proteins that are kind of holding these things in parallel orientation. So these are really important for bringing in the surface area, is important for bringing in carbohydrates and amino acids and lipids that we use for our energy source for the stuff that's going through our stomach, and the absorption is important to have this big area, surface area. So this is the microvilli, but then we'll look at the next part where it's kind of coming down into this terminal web. And these, there's a lot of that cross-linking. Calmodulin, for example, might be cross-linked here to kind of hold this all together and form a structural kind of component of the whole cell and in these microvilli locations. Actin is also very associated with the membrane. So that kind of forms the backbone, the structure of the membrane. You can see this kind of membranous area here that's shown how this is kind of attached at these little locations, and it's kind of holding this up. I don't know if you've ever looked at how a kind of modern house is organized, but you kind of have the wood that's kind of put up, the framing, and then you put drywall over it. So I kind of think of these as like the framed wood, and then this, the lipid on top, that's kind of the drywall that goes on top of it. So you have those kind of two different layers. And so this is really important for holding the lipid membrane in place, and then connects to the inside to the outside and connects everything. Or you kind of think of it as like the roof of your house or the outside of your house, but it kind of holds it in place. And I want to go back to another figure that we looked at earlier in a different show that where we looked at the different components here. So you might remember that tubulin structural protein that we talked about earlier. This is the actin, and you kind of see that it makes this little square, but it really outlines the outside where the lipid membrane is, and it really defines the borders of the cell. So this is just an image of a cell, and we're kind of looking at this cartoon and comparing it to this cell and why the cartoon is drawn the way it is. There are those different microfilament associated proteins, the actin, band, spectrin, and curin. Those are going to be attached, associated with that kind of that structural protein, and then also tethered to the membrane. I don't want to spend too much time on signal transduction because we spend more time on that in the other biochemistry class, but there are some associated proteins that there are different signaling mechanisms that regulate these different stress fibers and how these microfilaments are organized. And so, for example, if we have activation of the Rho GTP through GEF and GAPS, we can actually activate the remodeling based on signal transduction mechanisms. And you can see where we would, in order to restructure, you need GTP, and to have GTP in the system, you have to have a lot of energy. So if you starve the system, it's not going to be doing much remodeling. If you have a lot of, if it's activated through the signal mechanism, you can see these kind of lights up with these stress fibers. And you can also see that if we activate this RAC, it's a different signaling mechanism, or CDC42. Those have different influences on the structure of this of the cell and the cell shape. Those different signals will activate the release of these different types of proteins that regulate the actin formation and structure. So these basically these things that can cut, things that cap, things that can polymerize or deactivate or break down these different actin microfilaments are all part of those different pathways that are regulating the structure. One really interesting thing with actin is that there is a Listeria monocytosis adaptogenes that is actually involved in it actually uses the actin microfilaments in its basically to hijack our systems for making us even more sick. So you may have heard there was, this is a while ago now, but there was a possible hummus listeria risk. So it had some of this bacteria in it, and then it can make you pretty sick. And what's actually happening if you would ingest this and this bacteria got into the host cell, what happens is it breaks free of the lysozyme, and then it will actually polymerize the actin within the cell. And then it can actually polymerize and kind of make a little rocket and shoot into the adjacent cell and get into there, and then it can get released and then cause some more damage. And so then it causes this major concerns. Their symptoms are fever, diarrhea, muscle aches, headaches, stiff neck, loss of balance, convulsion. And it's also really, really dangerous in developing embryos. So if you're pregnant, getting listeria can be really, really damaging to the baby. So that's why it's actually recommended that pregnant women do not consume lunch meat without microwaving it first, because that would kill the living bacteria. Also things that like the hummus, for example, some of the things that can hold on to those bacteria can be dangerous and should be avoided by mothers. But you can see over here, this is a fluorescence imaging. You can see the actin outside of the cell in the green, and then red is actually the bacteria, so listeria, that is labeled. And then you see these little bacteria, like these little kind of rockets shooting off. They polymerize that actin and kind of shoot, and they made it into this adjacent cell, as you can see down here. So that's actually what's happening. It's polymerizing the actin to kind of shoot into the next cell over and cause that damage. So this was above here, the microtubules we looked at before, and here are the set for the microfilaments, cytochalasin D, latriculin A, phalloidin, and these again are all coming from different types of other living creatures, fungal metabolite, a red sea sponge, another fungus, the death cap fungus. These are all doing something, so preventing addition of monomers or sequestering actin, binding, stabilizing microfilaments, so they can cause major problems messing around with those in our cells, as you can see. The last type of filament I want to talk about are the intermediate filaments, or IFs. And these are not found in the cytosol of plant cells, but they're really abundant in many animal cells. And a typical, common, abundant intermediate filament is keratin, and it's a really important component of structures that grow from the skin in animals, like fingernails, for example, or some horn structures. Those are really important in some of the compounds that are part of those more ectodermal tissues, for example. Their intermediate filaments are the most stable and least soluble components of the cytoskeleton. So they likely support the entire cytoskeleton that we're looking at. What can be really helpful for intermediate filament proteins is that they're really tissue specific. So if we want to, for example, if we're looking at cancer and we don't know what type of cancer it is or what that cell is supposed to be, usually there'll be an expression of where it came from. Usually we can figure that out because it will express a certain intermediate filament protein. So that is what can define or help us figure out where it comes from. So that's also helpful for microscopy or doing some experimentation to understand what type of what is the cell developing into. For developmental biology, that's really helpful because we can identify the cell type based on the intermediate filament proteins that are being expressed. So that can be really helpful. They're usually grouped into different classes, and then that's helpful for us to kind of break down what type of cell component we're talking about. So here are the different classes that we can see, and the different types of tissues. So if we're talking about class 1 or 2, keratins, usually we're talking about epithelial cells, usually involved in mechanical strength. Vimentin, looking at fibroblast or mesenchymal cells, or the lens of the eye, for example, helps to maintain the shape. Desmin, looking at muscle and smooth muscle cells. So smooth muscle is kind of the contractile muscle in your stomach, for example, not necessarily your skeletal muscle. Glial cells, or astrocytes, we use a glial fibrillary protein, GFA. So that's a specific component that's only found in astrocytes. And then we'll have different axons and neurons. We'll have different other types of intermediate filaments that are expressed depending on the long, medium, heavy chains of these different neurofibrillary proteins. So these are different types that are expressed in different types of neuronal types of cells. We have nuclear lamins, and these are in all types, so this isn't specific for any of these above. Neuronal stem cells, nestin, this is all a precursor. Also sometimes nestin is expressed in pre-muscle cells as well, myogenic cells as well. So these are, we can use this information, and it's really helpful in developmental biology to understand what types of cells we're going to turn into, or what types of cells we're creating if we're looking at development in a dish, for example, or during a developing embryo, for example, to identify what is going to turn into what. And this is an example for how this can be used. And sorry, this is a little bit blurry, but it should still kind of get through the kind of the point. We have intermediate filaments, and then we have different types of during development. We're going to turn into keratins, or we're going to turn into different muscle cells, the neuronal cells, the nerve cells, different types of laminins. So these can all help to distinguish where we can turn into and what types of cells and where they are as well. So we have the intermediate filaments can be cytoplasmic or nuclear and have different functions in those areas. Even though there are different types that are typically cell-specific, there are fundamental subunits of these intermediate filament proteins and how they're organized. So they are typically dimers, and they're fibrous rather than globular, which means that they're kind of more elongated in shape rather than kind of blobby. In this figure, you can see that they each has a homologous central rod-like domain in the middle. It's kind of a big, longer chain of about 310-ish, 320 amino acids in length. And then flanking those domains are the N and C terminal domains that are more globular, if you will, on those ending endpoints. And the basic structure is you have this coiled central region of the intermediate filament proteins. And usually you can see there are going to be in parallel formation. So the C terminal and the N terminal ends will usually be on the same side. And then when they're aligned end to end and together here, usually you have those two different groups coming together. So you'll have the interaction of the filaments from end to end and then side to side as well. So these overlap and build on top of each other and create these structures. And in neurons, they're transported and easily remodeled, and they're a lot less susceptible to chemical attack. So there are not a lot of natural compounds that will mess around with the intermediate filament proteins. So if we look at one of them that is shown in this, it's a synthetic compound. So it's acrylamide. It causes loss of intermediate filament networks. So those things can be used to kind of mess with the structure. There was a story that there was a poisoned Harvard researcher that someone was angry with their primary investigator, and I guess they were squirting acrylamide in that person's coffee, and they got really sick, and it causes ataxia, so irregular movement, skeletal muscle weakness are usually symptoms of that acrylamide-induced central nervous system neuropathy. So it's certainly not common, but there are things that can influence the structure and the stability. Here's a scanning electron microscopy image where there's been kind of color overlay, so you can kind of see some of the different regions of the microtubules and the intermediate filaments, and some of the interacting proteins that kind of hold the structure of these things together. And so in addition to those structural proteins, there are always these kind of interacting proteins that help to glue and organize and regulate the regulate the structure and orientation between all of them. These intermediate filaments are really important for tension-bearing role and to support the structure of the cell. And they really confer a lot of strength to the tissue. For example, the axons need to have a lot of structural stability. They have really important that they're they're able to resist bending or compression, and they serve as an element that helps to contract and allow this to kind of maintain the shape, because this ability to have signal transduction from the cell body to the axon to the neuromuscular junction, for example, or the growth cone, is really important in our tissue. And neurons in particular have very elaborate cytoskeletal structure, where you'll have, for example, in green, you have microtubules. In purple, that you can't really see very well, but purple, you would have the intermediate filaments in the middle, and then in red, we have the actin microfilaments on the outside and the axonal growth cone. So they're all really important for the function of our cells and our bodies, and there are a lot of interesting things that we can learn about these cells because of these types of structural protein. And that is all I wanted to talk to you about the structural proteins for this chapter, and hopefully you've learned a little bit about that, and we'll next get into our case study to look more closely at how these structural proteins are important for function.